Hi, hello and welcome. Microbehunter here, Oliver that is, and today I want to answer another question from one of my viewers. This comment, or this question rather, was posted on a YouTube video. It goes like this: if all cells of a human being have a complete copy of the respective genome, and then there are two questions: how are all the cells so different? And number two, how do they all work together so effectively? So thank you very much. It addresses a very central question in biology. And the question is I'm going to formulate this now a little bit differently, is if all of the cells of a body, of an organism, let's say the human body, we all came from one cell originally, a fertilized egg cell. And by cell division, the DNA is copied first, and then by cell division it's passed on, and the cells divide, and then we have a multicellular organism. Now, why is it that even though all of the cells have the same DNA, that there are so many different cell types around? I mean, shouldn't the cells all be the same if they have the same DNA? So this question addresses a paradox, and I think that's a very central question, a very interesting question, and I'd like to quickly answer it first very briefly, and then a little bit in more detail later. The short answer is that even though all of the cells have the same DNA, not all of the cells use all of the DNA at the same time. So this means that, to give you a concrete example, you've got skin cells, for example. They also carry the DNA to produce certain, let's say, digestive enzymes which are needed for digestion of food, but this DNA is switched off. Okay, so it is not used. As a matter of fact, this is called, if you look, for example, if you use a microscope, and if you look into a nucleus, you can actually see two types of chromatin, two types of DNA, the so-called the euchromatin and the heterochromatin, because the nucleus, especially under the electron microscope, does not have the same color, but there are some certain regions that are more dense, and those regions that are more dense has more highly packaged DNA, and this DNA is not active. Okay, so what happens, what the cell does is the cell switches on certain genes, certain DNA regions, and as a consequence, these DNA regions that are switched on will then start to produce proteins, and these proteins that are then produced determine the characteristics of a cell. Okay, so that is kind of the short answer. The more elaborate answer now is the following: researchers have now found out that while it is the textbook knowledge that all the DNA of all cells are the same in a human being, actually it's not like this. They're now finding that there is more and more DNA variation within the cells. I want to give you one concrete example here as well. For example, the white blood cells, some of them produce antibodies, and those antibodies are produced randomly. Okay, so this means the shape of the antibodies is there's a random variation, and in order to do that, the cells introduces deliberately mutations and randomizations of its own DNA. So different white blood cells in that regions have different DNAs because they have to produce different antibodies. So it's a rather specific example, maybe, but I just want to make clear that overall, while the textbook knowledge that all cells of the same organism have the same DNA, if you look in details, it's not the case. However, those variations, those small variations, are also, of course, not enough to explain why there are so many different cell types around, and that can be explained by the so-called selective gene expression, by switching certain regions off and certain on. And how does this happen? This can be controlled chemically, and if certain factors are present, then certain regions of the DNA will start to become active, and then a substance called messenger RNA is produced, which then codes for proteins. So this is kind of the central dogma of molecular biology. That's what they call it. The central dogma of molecular biology is that you have a DNA which codes for proteins, then it's transcribed to mRNA, which then passes on the information to the ribosomes, which then make the proteins. Okay, but I'm sidetracking again a little bit. Overall, it's like this: different cell types exist, different tissues exist, because the different cells start to express different DNA regions. In other words, different DNA regions become chemically active, and others are not active. And the thing is the following: so-called stem cells what they can do is those stem cells still have the possibility to form other cell types. So stem cells are still very general cells, and by adding the right substances, the right chemicals, theoretically you can direct them to form and to specialize into other cell types. And what scientists are now have been doing for a couple of years now is they've been partially successful in doing this. What I read in the news is you want to take one cell type, for example, a skin cell, and by adding the right chemicals, you want to reactivate the DNA in such a way that it becomes a stem cell, a more general cell, and then by adding again the right chemicals, you change it so that it becomes a nerve cell. Okay, so this would be kind of the idea behind reprogramming in a cell, and this also, and because this is indeed possible to a certain extent, this actually shows that you can switch on and switch off certain DNA regions. And now to the second question that you've asked, is how can the different cells cooperate so well with each other? That is, I think, a more difficult question to answer. Overall, it's also like this. There's a lot of chemical signaling going on between the cells. And there are certain bacteria or certain, it doesn't even have to be bacteria. I think even certain protists also do that. What they have is while there are single cell, they can, by chemical signaling, they can cooperate and they can actually form something like fruiting bodies. So I think they must be the slime molds. That's correct. Now I remember. The slime molds, they are able to, some of them are able to do that. There's chemical signaling going on. Even though these individual cells, they are able to cooperate in such a way that they form the first structures that kind of resemble like something like a multicellular organism. This works by chemical signaling. And a few years ago, and even right now, scientists are doing this, is kind of ethically problematic, I think, is what you can do is you can take the cells, the stem cells of one organism, and inject them into an embryo of another organism, and then they will actually be incorporated and they will cooperate. Okay, so this actually does work. They have done this on mice before, and these were not related mice. These were quite different. Okay, genetically very different. And what you had is a so-called chimera, a hybrid chimera mouse. A chimera mouse, that's what they call it, is because some cells were from one mouse and the cell next to it were from another mouse. So it was kind of like a patchwork organism. This is it also works. So even though the cells are very different from each other genetically, even this works. Okay, so it is kind of interesting that there are this cooperation seems to work if you mix the cells at a very early stage. If you have, for example, now a full-grown organism like a human being, and you do an organ transplant where then you know that you have the problems of organ rejection. But this does not seem to be such of a problem if you mix the cells in a very early stage somehow. Okay, so this is a very brief, very brief answer to your question. They work together because the cells of different body cells work together because they are chemically signaling each other, and they have, they were not thrown together at the end and work together, but they have developed during the development of the organism, during the lifetime of the organism, they have established this codependency on each other. Okay, so thank you very much for the question. If anyone has any other questions, please send me an email, and I'll try to answer them. And of course, you can also leave comments here. All the best, bye bye, and happy microbehunting as always.